Today I'm going to talk about zoonotic diseases and how globalization has kind of affected some of these and caused some of these diseases to proliferate. So the reason, one of the I guess inspirations for this talk was this book. It was a book that Dr. Mai, my old internal medicine program director, gave me on graduation, and I think she thought it was supposed to be like kind of a fun read, and it was really interesting. He talks a lot on a medical level as well as kind of on a layman's term, and he tells the history of these diseases and these stories, or these diseases and how they evolved in more of a story format. So it's really interesting if any of you guys are interested in learning more about zoonotic diseases. All right, so to start with, we have to kind of could back up to what the definition of a zoonotic disease is. So according to Mandel's, zoonotic disease, or zoonosis, is an infectious disease of humans that originates in animals. So very simple definition in that aspect, and we can apply it in different ways. So when thinking about zoonotic diseases, it's important to remember how they got from the animal to the human. So there's a few different ways of transmission, and I think we think about these a lot in the specific life cycle. For example, when we're talking about Zika or some of those diseases, we know that there's a way for it to for the disease to get from person to person or from animal to person. But if you think about how it ever got from animal to person, you can back up and look at some of these processes. So the first one is transmission from a live animal reservoir to a human, and so this is when the animal is still alive and the virus is in some way gets into the living human. There's also dead animals or reservoirs that infect the human. So this is often seen in meat reservoir, bush meat, or transferring viruses from the animals to the humans. There's also reservoir that use intermediate hosts or amplifying hosts, and we can see this. We'll see this example a few times in pigs. And then there is also reservoirs that are vectors, and we think about these with our mosquitoes when we talk about malaria and Zika and some of the other flaviviruses. And so one of the things when you're thinking about if you know that the virus can go from an animal to a person, that's one type of transmission. But then it's also important to look at some of the diseases that we know have caused pandemics or affected a lot of people. And the viruses are also not just capable of jumping from animal to human species, but also going from human to human. So different modes of transmission, such as sexual contact, respiratory, iatrogenic, as we see with medicine occasionally. So when we're talking about, throughout this talk, we'll talk about a few different types of infections that can transmit, and mostly we're going to focus on viruses. So this topic is pretty big when we talk about zoonotic diseases, so it would be impossible to cover all of this in 1 hour. It would probably take a whole semester of class to cover all the different zoonotic diseases. So today we'll mostly focus on a lot of the viral zoonotic diseases. And when we kind of break down how the viruses are so successful at being zoonotic diseases, we can look at them between DNA and RNA viruses. So, and I'm making this pretty simple. We all know that there's double-stranded and single-stranded DNA viruses, and also single and double-stranded RNA viruses. But when you look at some of the basics between the DNA and RNA, you can understand why the RNA viruses are typically more successful at becoming epidemics or pandemics. So when you have your DNA virus, they're usually double-stranded, except those exceptions we were talking about. Because they're double-stranded, they're usually more stable. And the reason that they're more stable is because they have a DNA polymerase, which it essentially proofreads the replication process. So anytime there is a A or a nucleotide base that is put down incorrectly, the DNA polymerase can proofread it and correct it. So of course this process is slower because of the proofreading process. RNAs are usually single-stranded, other than RNA viruses are usually single-stranded, other than those exceptions too. But they have a higher mutation rate because they don't have this DNA polymerase that is constantly going back and checking the virus. So you see a lot more errors and essentially mutations in the RNA viruses. So as we talk about the viruses today, kind of keep that in mind and how we're going to get the virus from the animal reservoir to the human. So when we think about how these viruses have evolved, we have to consider what has happened in globalization and how humans have kind of essentially caused some of these pandemics. And so the World Health Organization has a really a detailed definition of globalization. So they say that globalization, or the increased interconnectedness and interdependence of people's countries, is generally understood to include two interrelated elements: the opening of international borders to increasingly fast flows of goods, services, finance, people, and ideas, and the change in institutions and policies at the national and international levels that facilitate or promote such flows. Globalization has the potential for both positive and negative effects on the development of health. So that's kind of a lot of words to, I think, describe that there's a connection between different countries and flow and people and ideas, and really all of this can affect disease, affect healthcare, and our policies and politics also affect these epidemics. So one thing to I wanted to back up and focus on a little bit too is that a lot of the diseases are affected by us and our ability to have transportation that we can get kind of across the world and essentially be capable of spreading a disease within a very short period of time. So if we look at this timeline, we can connect a lot of our pandemics that we know of and also relate them to the progress we made in transportation. We can also connect it to the kind of two big events where a lot of these pandemics happened, was World War I and World War II. So keeping that in the back of your mind too, a lot of transportation was either increased due to the movement of soldiers or decreased due to the amount of goods and availability that there was in the world at that time. So in 1903 was the first flight, which occurred by the Wright brothers. So I have to include that because it's from my home state in North Carolina, and we fight with Ohio a lot, but we are the first in flight. And then after 1910, there was a lot of different aircrafts that were made and developed, and a lot of this was in preparation for making fighter planes for World War I. But it wasn't until 1919 whenever we had commercial aviation, and these were just small little, like 5-passenger planes at this point. So they weren't transporting what we currently know. In 1927, there was the first transatlantic flight by Charles Lindbergh, and that's his little plane, model of his little plane down there. And then in 1932, Amelia Earhart was the first woman to fly across do a transatlantic solo flight. And then in 1933, we had really what we more know of as commercial airplanes. So Boeing made a 247, and then in 1938, we had our first pressurized cabins. And we continue to go on throughout World War II. Some of the commercial progress in commercial jets were delayed until after World War II. And then in 1949, we see some of those, actual jet planes increasing, but it wasn't until 1958 where we really started to produce them, and then Boeing had the 747 in 1960s. So if you kind of go through all that, there's a lot of the diseases today that we're going to talk about that went through that timeline and were affected by all of this. And as you can see, their aviation does have an effect on the transmission of infections. This was a flight map that I did on Saturday morning at 8:45 in the morning. So all those planes were like currently in the air at that point, according to the flight tracker. And they estimated that there's greater than 100,000 flights per day, or 10,000 flights per day, and 36.8 million flights occurred in the year of 2017. So if you can imagine, if there's just a small infection on one of every of that plane, and you transmit it to a different location, you're going to spread a lot of disease. So things that also to keep in mind that can spread disease through transportation, specifically with planes, is that during travel, passengers are at increased risk for infection due to the high density of the layout and the seating, so everybody's close together. There's low humidity and hypobaric oxygen, and recirculation of oxygen, I didn't include that there too, that also increased the likelihood of transmitting infections between the passengers and the plane. Okay, so the first one I want to talk about, I think there's an important lesson to discuss with smallpox, even though smallpox is not a zoonotic disease, so I want to make sure that I made that clear, but it has a few educational points that I think will be good to kind of carry forward. So this disease does demonstrate how globalization affects the spread of disease, but also teaches us an important lesson on eradication too. So the Great Southeastern Smallpox epidemic occurred in 1696 and lasted till the 1700. And it began in 1650. The Virginia traders ventured into the Carolinas, and that's kind of how they followed the river patterns and created their trade routes. It wasn't. They think that the smallpox arrived through the African slave ships and then was able to be transmitted through those trade routes. And most of the trade routes were stimulated by either slaves or rum trade. Those were the key players that were thought to assist with the spread of smallpox. So this is a map that kind of shows the spread of smallpox. So the black arrow is the trade routes, and then the dotted arrow is where they think smallpox had spread. And the little plus signs are where they had a depopulation. So a lot of the Native Americans were affected by smallpox and caused a lot of deaths during that period of time. So in 1980, the World Health Organization declared that smallpox had been eradicated. So eventually we were able to create vaccines and eradicate smallpox. So the diseases something that they focus on when they talk about creating vaccines in order to eradicate diseases is that most of the diseases are not zoonotic that we're able to create vaccines for and that we've been able to eradicate. Having an animal reservoir can lead to recurrent outbreaks of disease, and what makes these diseases so unpredictable and difficult, that's what makes them so difficult to treat and prevent. So the first one that's a zoonotic disease that I want to talk about is yellow fever. So yellow fever is a flavivirus. It's present in the tropical South American and sub-Saharan Africa, and it typically presents with fever, headache, and myalgias, and begins abruptly, accompanied by conjunctival injection, facial flushing, and relative bradycardia, the Fauché sign. And eventually it leads to icteric hepatitis and hemorrhage. So one way they say you can differentiate this too from some of the other hemorrhagic viral infections is hepatitis is checking for albuminuria. Okay, so the yellow fever was known as the American plague. It first appeared in 1648, and the first definition or definitive evidence of yellow fever in the Americas was in the Mayan manuscripts, and it was suspected at that time to be imported from West Africa through trade ships, similar to with the trade ships as some of the other viruses. in 1668 through 1699, the outbreaks occurred all throughout the East Coast. But it wasn't until 1848 that they realized that there might be a vector, and the vector was probably mosquitoes. So at that point, it was 1898, so several years later, it started causing problems for the U.S. Army. And that's when Walter Reed got involved and assisted with trying to eradicate yellow fever through eradication of mosquitoes. So in 1900, the Reed Yellow Fever Commission proved that yellow fever infection is transmitted to humans by the Aedes aegypti mosquito. And then 1905, after doing that and focusing on eradication measures, we were able to eradicate yellow fever. 1930, in 1930, there were 2 yellow fever vaccines that were developed, the 17D vaccine and the French neurotropic vaccine. So when we're thinking about yellow fever, there's 2 different life cycles to keep in mind, and this can be applied to many different zoonotic diseases. We talk about it with Zika more recently, but knowing the 2 different life cycles, the life cycles, the jungle and the urban. So with the jungle, this is kind of the natural life cycle of the mosquito affecting the monkey then getting bit by another mosquito, and the life cycle continues to spread the disease. When a human is affected is when that mosquito that's part of that kind of jungle life cycle bites a human, and then that human goes into the urban population, and there are appropriate mosquitoes that are able to spread it from human to human. And then there's also the intermediate or savanna life cycle too, which usually just occurs in Africa when there's very close populations of both animals or monkeys and humans in the same environment. So that discusses yellow fever. So the next one is the flu. So there's a lot of different things to talk about with the flu. So the 1918 Spanish flu is probably one of the most well-known pandemics and zoonotic diseases. So in January of 1918 through April of, not 2019, that's an error there, 1919, the Spanish flu affected the Americas, and it really became a pandemic. The origin is unknown. However, there's several different theories that exist on how influenza originated at that time, and probably the most well accepted is that it started in Kansas, where there was a high population of hog farmers that also lied within, in that farming area, there was a bird migratory pattern. And so at that time it was proposed that because of the hogs and the birds, the native birds, that there probably could have been the zoonotic disease could have been kind of spread. So when in Kansas, it was proposed that it was transmitted through France by soldiers in World War I. So basically it went from Kansas, and you can see those kind of lines up there, the red lines, over to the eastern side of the United States, and then because of the soldiers traveling across the globe because of the World War, eventually going into Europe. But it didn't gain much of attention until it affected the Spanish king, and when he contracted the illness, he became known as the Spanish flu. So it's kind of a misnomer. It wasn't first. It's not originated in Spain. So in September of that year, it spread by Navy ship to Philadelphia. So once it got into Philadelphia, they had noticed that a lot of the soldiers were sick, and they expected them to have this influenza virus. But there was a big parade, the Liberty Loan Parade, that was planned for later in that month on September 28th. So the city decided to continue with the parade. However, many of the people that attended the parade ended up becoming ill, and they ended up having 12,000 people in Philadelphia that ended up dying because of the influenza virus. And it's proposed that because of the parade, it spread more rapidly. So interestingly, with the Spanish flu, it had 3 different waves. So there was the first wave, second wave, and the third wave. And the second wave was the more deadly wave. This graph represents the deaths in the UK, just because they had the best, at that time they were keeping the best data, but it's proposed that that's kind of how the wave was in the Americas and throughout the rest of Europe. So it's currently still the deadliest disease outbreak that has been known, and it killed 500 to 100 million people, it's estimated to have killed. So when we're talking about influenza, so now we know kind of how deadly and powerful this disease was, but to understand why this occurred, you have to back up and know a little bit more about the microbiology of influenza. So there's two different important parts of influenza, so hemagglutinin and neuraminidase. So these are both surface proteins or spikes that are on the outer surface of the influenza virus. So there's 16 different types of hemagglutinin and 9 different types of neuraminidase, and these lead to the different types of influenzas that we kind of know about today. So the big shifts are whenever we have reassortment, and then the smaller ones, the antigenic drift, is when we have minor mutations in these viruses. So humans typically are only affected with H1, 2, and 3, but H9 and H5 have also been known to affect humans, but so far when they do affect humans, they're not known to have human-to-human transmission. So kind of going back to where the influenza virus originated, we now know that waterfowl is a reservoir for the influenza viruses, and as you can see over on the left-hand side, the influenza kind of lives within the waterfowl, and all of the different types of hemagglutinin that are found on the influenza have been found in the waterfowl. They can go from domestic into domestic birds, and sometimes they make them ill and sometimes they don't. But the swine or the pig has been known to be the mixing vessel for the viruses. So essentially, the viruses will go into a form that affects the human and affects the birds, can go in, both infect the swine, and that's how we get the new different strains of the flu, and therefore causing something that the human population hasn't been exposed to, which would be more of the antigenic drift. So the biggest topic I think we're going to talk about today is HIV. So this one, we'll talk a little bit about the discovery of HIV first. So it was first discovered, and actually I wouldn't say it was discovered, but there was awareness in 1981 that about a cluster of cases in UCLA that the patients all had PJP pneumonia and Candida infections, and were noted to have depleted T cells. This was reported in the MMWR in June of 1981. So this physician, he noticed all these different cases and realized that there was something alarming of this, and had it posted in there. It wasn't until 1983, and there were several clusters after that. But we'll go into all the details. But it wasn't until 1983, though, that Montagnier and Gallo, he was in Paris and Gallo was in Bethesda. They both published at the same time in Science magazine in May about a virus that they discovered that was associated with this clinical presentation, and it was essentially a syndrome at that point. So between 1981 and 1983, there were a lot of different names that had been kind of given to the virus or given to what they thought was a virus or the syndrome that was associated with it. But this is the first time they actually identified something that could cause it. And they each had their own name for the virus. So one was lymphadenopathy virus, or LAD, and then Gallo named it after the other HTLV viruses that were studied, and he named it HTLV3. In 1985, importantly, Conkey and Essex discovered SIV. So at this point, the question was, where did this virus come from? So people started looking into monkeys and chimpanzees, and because they had noticed that some of their monkeys were having similar illnesses in the labs. So they discovered SIV in 1985, and they published their work in Science. And they discovered this in the macaque monkeys that they had in a laboratory. So SIV was later found to be in 3 different monkeys: the African green monkeys, the rhesus and the sooty mangabeys. But it wasn't until 1989 that somebody had really established an association between HIV and the SIV, those viruses. And this was Hirsch and Korb. So they established that between, and this was HIV-2 actually, rather than HIV-1. And they proposed that SIV in an African sooty mangabey infected a human, resulting in HIV-2. So they made a pretty bold statement in their paper, kind of creating this relationship between the 2 viruses, and at that point identifying HIV as a zoonotic disease. So that's like a super brief history of what kind of happened between 81 and 89, but some of the big points to touch upon. So the question at that point, once it was decided that it was, or a common thing that was talked about, once it was decided it was, or thought to be, a zoonotic disease, how did it get from the reservoir monkey or chimpanzee to humans? So there was a few different theories. One was the cut hunter hypothesis, so that a hunter that was hunting these monkeys was directly contaminated from the animal to the humans during preparation of meat. The other one, which I thought was interesting, is the oral polio vaccination theory, which there was a, they were promoting polio vaccination in Africa in 1958, and we'll come back to that in a little bit. And then the third proposal was that hypodermic syringes could be contributing. And in particular, there was urge to eradicate trypanosomiasis and other venereal diseases. And there was a clinic that was giving medications to eradicate these venereal diseases, and specifically they were doing this for a lot of the sex workers in Africa. So in 2006, so that kind of, keep that in the back of your mind while they're trying to figure out how it did get into humans. So in 2006, using genomics, Keel showed that chimpanzees from Cameroon carried a very similar SIV to HIV-1, therefore suspecting that they were the reservoirs. So we kind of established earlier that we thought that there was a certain strain of SIV that was associated with HIV-2, but they didn't know until 2006, or really connect until 2006, that SIV and HIV-1 were connected. In 2008, there was an analysis of preserved blood samples and lymph nodes that revealed 2 different sequences. So there's a ZR59 and a DRC60 is the names that they just gave them. And using genetic trees and time models, they were able to predict that HIV-1 was circulating as early as 1908. So that's a lot earlier than if you go back to when we were talking about it presenting in America in the 1980s. This is quite a bit earlier. And it also, that genetic tree and the kind of dating it back to 1908 also had another important interest, because there was still the question of how it ended up getting from from animals to humans, and that there was a lot of pressure on the fact that the polio vaccination could be contributing to it. But the polio vaccination did not occur until 1958, so this kind of cleared that theory of being the cause of the zoonotic disease. And so here is a map that shows what the proposed transmission was. So looking at one, this is where they were saying they're labeling it as the butcher of the chimpanzees, but of course we don't know that that was definitely what occurred. following the river where the blue dots are, going down to number 2 along the river, and then going to the city of Kinshasa, where the proposed epidemic of HIV began and then spread throughout the world. So in Kinshasa, there were only 10,000 people in 1908 predicted, and it was a very isolated town. But in 1898, a railroad was built into that area, and by 1940, the population increased to 49,000. So to kind of think about globalization and how it could affect disease, introducing modern transportation into that city could have contributed to the disease spread, eventually leading to disease spread to America and Europe and the rest of the world. Okay. And this is just to kind of represent that once the disease was present, it did transmit. It may not have made itself known until later into the 1980s, but through looking at different genetic models, they have traced it back to different areas. It's thought that in 1966, HIV made its way to Haiti, and then between 1969 and 1972, that's when it probably traveled into the Americas. And then about 10 years later, when we started seeing the syndromes that we currently identify as HIV. So the next one that we'll talk about is SARS. So this one is one that I think a little more modern. In the last 10 years, I remember being in high school whenever this one was occurring. The first case was reported in November of 2002, and it was associated with a province in southern China. It's thought that the reservoir is probably the horseshoe bat with an intermediate host of civet cats. That was the original thought. They identified it in both of these animals. However, it's not been kind of clearly proven that it is this bat. Humans have become the host due to interaction. It's thought that humans have become affected due to interactions in the wet markets of China. So the wet markets became more popular during this time in the search for more exotic foods, and then because of having these animals within those markets, there could be increased likelihood of having a zoonotic disease develop. As we said, the first case was reported in November of 2002 in China, but the reason that it spread so quick is because there were a lot of what they called super spreaders, including a man that was connected to 60 cases that affected a hospital. So he presented into the hospital. He affected many healthcare workers, essentially all healthcare workers. And that physician then later went to Hong Kong for a wedding, where he himself became a super spreader, then spreading it to many hotel guests. And then those hotel guests were kind of went back on to where their home locations were, one of which was in Canada, Toronto, Canada, and that's how we kind of got from November of when they think that it originally began in southern China to February of 2003 when SARS had kind of become a worldwide virus at that point. The one point that made it so capable of spreading was that it was spread through respiratory droplets. Once it made its way to humans, it was able to spread from human to human through the droplets. And so this was the paper that kind of claimed to be the discovery of SARS. So this was by Nichols, and in a paper called Clinical Virology and SARS, the Clinical Virology and Pathogenesis. And it was published in 2003 in a journal called Respirology. But there was criticism at that point of that this was a little bit delayed of recognition of the virus. And a lot of it was thought to be due to a lack of communication between Hong Kong University and the Institute of Respiratory Diseases, which was located in China. And a lot of it was due to some of the politics that were going on between China and Hong Kong at that time. So the next one is Nipah virus. So this was discovered in 1998. It's a febrile illness with encephalitis. It was first noticed in Malaysia. So at the same time of having the humans having the encephalitis and respiratory problems, they noticed that pigs were also getting sick, and they called it the barking pig syndrome. Originally, they thought that this infection was Japanese encephalitis, but it was later found to be a new paramyxovirus that they discovered when they did CSF analysis. In order to eradicate the disease, they had to kill off over 1 million pigs, which caused a lot of loss of money and was pretty catastrophic to a lot of pig farmers in the area. But in total, there were 276 cases reported, and 106 were fatal. And no further cases have been reported in Malaysia at this time. The next outbreak was in India. So this occurred in 2001, and there were 66 humans at that time. They're not sure that this was actually how the zoonotic event occurred at that point. So they didn't have the same connection with the pigs as they did in Malaysia. And then in 2007, there was also an outbreak in India, but this was only one person that was infected by their ant, and it resulted in 4 human-to-human transmissions. And at that point, it was known to be 100% fatality. So the place that Nipah virus still seems to be a problem is in Bangladesh. So there have been repeated outbreaks since 2001. And they have associated this with the date palm sap that is often they collect the date palms in the winter months. But the bats at that same time period have been known to drink the sap on the trees and cause human to. The thought is that because the bats would be the reservoir, causing infection of the sap, and then the humans are able to or come in contact with that sap, and then you have the process of creating the zoonotic disease. The other thing that makes it so important, as we were discussing at the beginning of the lecture, is that it also has human-to-human transfer. So in 2004, there was an outbreak, and 94% of the cases at that time were reported as human-to-human transfer. So as I kind of mentioned, the reservoir for Nipah virus is thought to be the flying fox, and this is a type of fruit bat. Does anybody know what the other another virus that the flying fox is a reservoir for? Ebola, right? Well, it's not proven. Yeah. Hendra virus is another one. Hendra virus is a virus that affects horses, or has been shown to affect horses in Australia. And so they did a kind of, these two viruses have been associated with the flying fox. Okay, so Ebola, we obviously can't cover this. It's a huge talk in and of itself, and all of these topics were. But we'll just kind of briefly discuss a few things. So prior to 2013, a lot of Ebola outbreaks were sporadic. It wasn't until then that that a lot of the kind of pandemic, as we're aware of it today, had occurred. So there's 5 different types of Ebola strains that were discovered between 1976 and 2002. The search for the reservoir is still ongoing. So as we mentioned, the thought is that it occurs in bats also, but there's been no definitive scientific evidence that has definitely linked it. They have shown antibodies in some of the viruses, or in some of the bats, but no definitive evidence. And then monkeys are thought to be, I think monkeys is what people traditionally think of as the likely reservoir, or what was originally thought, but they're really an accidental host. So there's 2 phases. The first phase, you have about 5 to 7 days of fevers, rash, joint pain, headache. Could be fever-like illness, or many of the other viral illnesses that occur in the areas where Ebola is present. The second phase is the more severe illness with GI tract abnormalities like abdominal pain, diarrhea, that can cause orthostatic hypotension, a lot of vascular system problems. It can cause you to go into a coma, CNS effects, and then the hemorrhagic manifestations that all occur later in the second phase of the disease. So I think why it became so important, I thought this is an older graph from when the epidemic was occurring in 2014, but it kind of shows how there were all these many outbreaks that dated back, but the 2014 epidemic was really where we saw a lot of deaths, and there were greater than 25,000 people at that point affected, and greater than 10,000 people died. So it's really not known why or when the actual event occurred. There's a lot of theories, and we could talk about them all day, or for a whole lecture about the theories on how the 2014 epidemic occurred. But a few kind of important things, I think, to relate it to globalization and why we became more aware of some of the things, or some of the effects of Ebola. So in September of 2014, the CDC confirmed the first laboratory Ebola case that came over to the United States in a man who had traveled from Dallas. And this is when there was a lot of hysteria related to Ebola, and a lot of awareness of what Ebola was from the general American population. In October of 2014, there was a healthcare worker at Texas Presbyterian Hospital who provided care for the index patient and tested positive for Ebola. So that was the first time that they had shown that the person that was came from Liberia infected an American healthcare worker. And then in October 2015, there was a second healthcare worker that ended up testing positive. But things were complicated by the fact that this healthcare worker had gotten on a flight on October 13th, and they had to isolate the plane and watch those people for a total of 21 days too, to see if anybody else was secondarily affected. So this is when we started, a lot of awareness in the United States was brought about to Ebola. So at that time, there were a lot of different news articles on if the Ebola was coming to the United States, and at that point it had a huge effect in Africa, but the news stories seemed to be related to what was going to happen in the United States, and should we allow the infected physicians and healthcare workers to come back into the country, and should we stop the flights and not allow people to come back. So I think this is when a lot of American citizens really became kind of more aware of what a zoonotic disease was, and had an opinion on some of the politics that did ultimately affect the spread of Ebola. As of January 2015, there were 8 doctors, nurses, or kind of aid workers that had been brought back and treated, and only at that point had 1 of them died. So that was kind of a whirlwind of zoonotic diseases. Does anybody have any questions or thoughts on any of them?